Okay, 3, 2, 1, it's jam.
What's up, science fan? Today we have a crash course on a very, very large topic. It's the cell. Literally, we're going to talk about everything about cells. We're going to talk about the cell theory, cell types, cell transport, cell division, and the cell cycle. Almost literally everything. We're not going to go into too much detail, but it's going to be an overview of everything you might need to know about cells going into your lives if you're not going to be a biomedic. So let's do this. The cell overview. So first things first, plants are made of cells. You can see in the background these are plant cells. You can see the chloroplasts in the cell walls. Animals are also made of cells. You can see that in the background there's red blood cells. You know that we're made of cells. And so generally the consensus that we can come up to is that everything that's living is made up of cells. And that is one of the tenets of the cell theory. It's a theory that ties all of biology together. And there's three basic tenets. And we're going to talk about who were the ones who discovered these tenets. So first we have Matthias Schleiden. He's a botanist. All plants and their products must be made of cells or products of cells. He concluded that by looking at plant cells. Theodor Schwann is a physiologist, and he had to study the parts and functions of animals and humans. He saw also under the microscope that animals are made of living cells. So all animals and their parts must be made of cells or products of cells, and so they had something in common. Their plants and their animals are both made of cells that have distinct nuclei and plastids. So they came up with the first two tenets of the cell theory. All organisms are composed of cells, and the cell is the fundamental unit of life. But there's a third tenet, and it's basically answering the question, where do cells come from? Do they appear out of nowhere? So it's from Rudolf Virchow, a pathologist. He coined the term omnicellula acellula, which means that all cells must come from pre-existing cells. But wait, some historians doubt that Virchow actually discovered it because he seems to have plagiarized his work from another dude named Robert Remarque. So this guy got plagiarized and then get credited because he's a Jew. So he studied chicken embryos and found them at different stages of division, and he figured out that probably they just all come from cells. So, you know, politics. But basically, the classical cell theory's three tenets are that all organisms are composed of cells, the cell is the fundamental building block of life, and that all cells must come from pre-existing cells. So this ties together all of biology and all of the different cell types. Speaking of cell types, what are they? So this is a type of cell that looks like a fudgy bar. This is basically a prokaryote. Before nucleus is the literal meaning of prokaryote, and that means that they don't have a true nucleus. So we have the bacteria and the archaeans. Your archaeans are also known, some of them are also known as extremophiles because they can live in extreme conditions. Not all of them, just a fair amount of them. So they look like bacteria. The one on the left is an archaean, the one on the right is a bacteria. So they look very similar. They have similar structures, but genetically, your archaean branch off closer to eukaryotes rather than to bacteria. So they're more related to us than to bacteria. Weird, right? So the cell types, aside from the prokaryotes, we also have the eukaryotes, the ones with the true nucleus. Okay? It's a cellular structure that contains the DNA. So the true nucleus can be found in plants, animals, and fungi. I don't think I need to explain what those are. Okay? So basically, we have five main cell types: plants, animals, fungi, bacteria, and archaeans. These also represent the five primary domains of life. Alright? So basically, cell theory links all of these five distinct domains of life together into one study, one branch of science. So the cell theory is one of the fundamental theories under biology. Without it, we wouldn't be able to know plants and animals are basically made up of the same stuff. So are bacteria, archaea, and fungi. Okay? So how can cells survive if they're so small? We have cellular structures or organelles that help them perform different processes. So different cells have different structures depending on what type they are and depending on what they need to perform. So we're going to focus on the plant cell, the animal cell, and the bacteria cell because fungi are weird and archaeans are very similar to bacteria anyway. So what holds the blueprints and instructions is the DNA. And the DNA in eukaryotes is in, of course, the true nucleus, which keeps the DNA locked up safe. So it looks like this. It's a giant ball with a nuclear envelope that is a double membrane system, meaning it has two membranes just to keep the DNA extra safe. The prokaryotes, on the other hand, have a nucleoid region, which is literally just a blob of DNA floating around near the center of the bacterial cell. So these are some of the features that they have for the nucleus. Again, we have a double membrane nuclear envelope with just pores that allow certain things in and out, but not the DNA. Your nucleoid region, on the other hand, has a bacterial chromosome, the main chromosome of bacteria, and plasmids, which are smaller rings of DNA, which they use to give certain genes to other bacteria or receive genes from other bacteria. So what protects the cell from the outside environment? We have the plasma membrane. All types of cells have a plasma membrane composed of a phospholipid bilayer, just two layers of what we call phospholipids. The cell wall for bacteria and for plants is an extra layer on top of the plasma membrane, and it's made up of different stuff between the bacteria and the plants. Okay, so in the plasma membrane, again, it's made of phospholipids, and it is usually characterized as being a fluid mosaic model, meaning it's fluid, it's wiggly, and it's a mosaic because it's not just phospholipids, there's a bunch of other stuff floating around. Your cell wall, again, let's just remember that your bacteria have peptidoglycan, a different kind of protein, in order to make the cell wall rigid, while your cellulose is the thing that makes your plants rigid. Cellulose is the thing that also makes it so that when you poop, your vegetables and fruits tend to come out the same way as when you swallow them. You can't digest the cellulose in cell walls. So what is the fluid inside the cell? We call that the cytoplasm. So just remember that inside the cell, we have the cytoplasm, and that's where everything else is floating around. So what gives cells their shape and form? We have the cytoskeleton. So just think if the cytoplasm and the cytosol are the fluid, the cytoskeleton are fibers in the fluid that keep everything in place. So how do cells have things that they can keep stuff in? What are the storage spaces? We have vesicles. It's basically a membrane bag. So if you take the phospholipids and you make a bag out of them, that's a vesicle. And we have bigger types. We have the vacuoles. In all cells, there are small vacuoles that are big storage spaces for water, food, or waste. And in plant cells, you have extra large central vacuoles that are even bigger than their nucleus. So it's very, very big to store water, since plants can't get up and drink water from water dispensers. And they also control what we call turgidity, or how hard a plant cell is. So that's it for the vesicles and vacuoles. For eukaryotes, you also have special things called lysosomes and peroxisomes. These are things that break stuff down. Lysosomes break stuff down using hydrolytic enzymes, meaning they digest stuff using hydrolysis. So it's necessary for both digestion and recycling of broken organelles, while your peroxisome breaks stuff down using oxidative enzymes, which makes it useful for breaking down fats or fatty acids. So they have different uses. Your peroxisome, also from the name peroxide, it can also break down hydrogen peroxide into water and oxygen. So they break down different things. So what if it's time to split? What helps the cell divide? We have the centrosome. It's basically just two paired churros that create stuff that can attach to your chromosomes and rip them apart during what we call mitosis. So the centrosome creates spindle fibers. What about movement? If the cell needs to move around, with cilia and flagella. Cilia are usually smaller and are more numerous, and usually they're not to move the cell around, it's to move stuff around the cell. So if there's stuff outside of the cell surface, it can sway back and forth so that it would move the stuff on the surface towards one direction. The flagella, on the other hand, is literally like a motor for the cell. So it's like a motor that rotates around so that it can propel the cell in a certain direction. So this is what it looks like. Your cilia, again, moving back and forth, and your flagella in a rotary motion. Okay. Next, where does the cell get its energy? We have two kinds of plastids for eukaryotes, all kinds of eukaryotes, whether plants or animals. They have mitochondria, the powerhouse of the cell. It creates ATP. On plant cells, on the other hand, we also have chloroplasts. So there's also some animals like the leech slug that has chloroplasts inside it. So your chloroplast basically takes the power of the sun and uses it to make sugar out of carbon dioxide and water. So both of them, interestingly, have their own DNA. So how does the cell process materials? We have a lot of stuff for materials processing. We have, for example, the ribosomes, which process amino acids to make proteins. So again, these are the protein synthesis machines. Without these, cells wouldn't survive. So all types of cells have ribosomes. Lipids, on the other hand, are made by eukaryotes using smooth endoplasmic reticulum. Your rough endoplasmic reticulum is embedded with ribosomes. So those ribosomes are what make it rough, and that's why it's for folding and transporting of proteins. On the other hand, your smooth endoplasmic reticulum, it transports lipids and detoxifies them. Okay? It doesn't have any ribosomes, so it just focuses on lipids. So how do we get these things where they need to go? We have the Golgi apparatus. It sorts, modifies, and packages all these different things into vesicles and then sends them either outside the cell or somewhere else inside the cell. Right? And that is basically all of your cellular structures. Your cell cycle, or the life cycle of a cell, on the other hand, is what happens during the general life of a cell. The common mission of living things is reproduction. So, yeah. If reproduction is the goal, cell division is one of the main goals of most cells. Again, most cells. So if it's going to split in two, it needs to prepare everything. And in order to prepare everything, we have what we call the interphase. So all of the kind of blue parts, G1, S, and G2, so most of the life of a cell is spent preparing for division. So that's the interphase. So if there's going to be two daughter cells, how much stuff does it need? Of course, it needs twice the amount of stuff. We have the G1 phase, or gap phase 1. This is entailed by the duplication of all the organelles in the cytosol so that the cell grows in size. So again, just double on everything so that they don't fight over it, like these two twins here. And there's a lot of conflict that happens within the cell. Sometimes there's a midlife crisis that goes on during the gap phase 1 or after gap phase 1. So we have the G1S restriction checkpoint. So between G1, gap phase 1, and the next thing, there's a checkpoint that checks whether the cell will go one way or another. So it asks the cell what does it want to do with its life, and it checks a bunch of things. So it can either go on to the next phase when it means to divide, or it can stop and go into what we call the G0 phase. So what is the G0 phase? It's a quiescent stage. It means that the cell has either felt like it's fulfilled its destiny and doesn't want to divide anymore, or it's given up because it's too far damaged and it's too old for it to function properly if it divides even more. So again, differentiated cells, meaning specialized cells that fulfill their destiny, will go into the quiescent stage, and senescent cells, cells that are too old, will also go into this stage. Now, if it does go into the next stage is the synthesis phase, which literally is just the replication of DNA. Your chromosomes, which are usually unduplicated, the ones that look like a single sausage with a nipple in the middle, will become looking like X things. Okay, so that's unduplicated and duplicated on the right. So if there's now double organelles and double DNA, what happens next? We have gap phase 2. In gap phase 2 is the synthesis of cell division protein. So it's preparing for division itself. But it seems like it's not super necessary because in certain species of frogs and in cancerous cells, they completely skip gap phase 2. So I guess this is just for the safety of certain processes in cell division. So the final prep stage to make sure that everything's ready is the G2/M DNA damage checkpoint. So it just checks if DNA is copied correctly, and if there's any damage, it will try to repair it, and if there's too much damage, it will go into senescence, apoptosis, or cancer. So we know what cancer is. It's the proliferation or the division of cells, super duper fast, uncontrolled cell division. Senescence is it getting old. So what is apoptosis? Apoptosis is programmed cell death. It's basically the self-destruct button on cells, and it's meant to reduce the risk of cancer. So it usually starts with the mitochondria, the powerhouse of the cell. It's like, imagine a Chernobyl explosion, kind of like that. So apoptosis has no cancer risk. That's why the cell sacrifices itself. On the other hand, if it continues to divide when there's DNA damage, it could lead to cancer. So if we make it past the checkpoint, is it time to divide? Basically, yes. So after interphase is mitosis and cytokinesis, which we are about to discuss. So coming out of interphase, your cell looks like this. It has duplicated chromosomes. The centrosomes are ready and primed to do their work, and everything's just there. So the duplicated DNA and the centrosome are ready. So in your cell division, we have mitosis. It's a form of somatic cell division, meaning body cells. So somatic just means body cells. So we have the acronym PMAT. These are the four main phases of mitosis: prophase, metaphase, anaphase, and telophase. Let's go through them. In prophase, we have a few different things that are happening. We have the start of the mitotic spindle. These are just fibers that attach to chromosomes. The chromosomes get thick, and the nucleolus starts to disappear. In the late prophase, your nuclear envelope is gone as well, and your chromosomes are fully thick. And then your spindle fibers are attached to the centers of the chromosomes already. So chromosomes are arranged by the fibers into a metaphase plate during metaphase. So metaphase is just when your chromosomes are lined up in the middle. So there's also a checkpoint here called the spindle checkpoint. So the cell checks if the spindle is attached properly to the chromosomes, because otherwise they're not going to do the next phase properly. So again, metaphase, we have the metaphase plate. If all goes well, if the checkpoint says all clear, then it's the anaphase time. It's the divorce of the chromosomes chromatids. So the chromosome is composed of sister chromatids, which are replicas of the DNA, and they're ripped apart by the spindle fibers during anaphase. And since the chromosomes split, now they have to be repacked, so there's the telophase. So in telophase, it's also known as reverse prophase, because it's basically the reverse of prophase. Instead of getting thick, the chromosomes instead of disappearing nuclear envelope, it reappears again. So it's just trying to pack things up into normal. And aside from packing things back up, we also have a need for the cell to actually split. So this is not part of mitosis. It's its own distinct stage. It's called cytokinesis. So for animal cells, they have cleavage furrows because it's just a dent. In plant cells, since they have a rigid cell wall, they just have the formation of a cell plate. It's a new cell wall in the making. So again, cleavage furrow for animals and cell plate for plants. Next, for sex cells, how do they get made? So we have meiosis. It's the way we make gametes or sex cells. So from diploid or two chromosomes, one from dad and one from mom, it becomes haploid, so that it's just N. So from 2N to N. So that when you combine the sperm and the egg, we have N plus N. The normal diploid becomes 2N. Okay, so what's different? How is meiosis different from mitosis? There are two parts to meiosis. You have meiosis 1 and meiosis 2, and each of them have their own PMAT, their prophase, metaphase, anaphase, and telophase. So what's different? What's new in meiosis 1? We have genetic variation. In meiosis 1, it has two processes that make sure that the sperm and the egg cell have unique genetic combinations. So these two processes are crossing over is the first one. So prophase 1 has crossing over. It's basically when your homologous chromosomes, or the chromosomes from mom and dad, they cross over, just like in real life, and then they exchange genetic material, just like in real life. So it's like a mix and match of the genetic information between your mom and your dad's chromosomes. Okay? So after the crossing over, in METS phase 1, there's also random orientation. So your homologous chromosomes, after they cross over, they're still stuck to each other. So they're paired up with each other, and they can be flipped. Either your dad can be on the left side, or your mom's chromosomes can be on the left side. So again, you can flip each pair of chromosomes. So it's like it's shuffling all of these chromosomes by pairs. So this is what random orientation is. So we have two processes. We have the crossing over, which is a mix and match, and we have random orientation, which is the shuffling of chromosomes. So that each combination, each daughter cell, each gamete has a unique genetic combination, and that's why you and your siblings aren't the same. So anaphase 1 and telophase 1 are basically the same as mitosis, anaphase and telophase. It's just the ripping apart and the repacking of the chromosomes. So the result is that after meiosis 1, we have two haploid cells with duplicated chromosomes. So if you look at this diagram on the left, we have two haploid cells, meaning we started out with 4 chromosomes here, but then at the end we have 2 chromosomes in each cell. It's just that they're duplicated, meaning they look like the X instead of the single sausage. Next, we have meiosis II. It's basically just trying to make the duplicate chromosomes unduplicated. They're trying to separate the sister chromatids. So it's just the same. Prophase, metaphase, anaphase, and telophase. As mitosis, again, exactly the same. There's no crossing over. There's no random orientation. It's just separating the sister chromatids. And the final product, we have 2 daughter cells from meiosis I times 2 daughter cells from meiosis II. We have 4 daughter cells in total, and they're all haploid and unduplicated. That's your sperm and your egg cells ready for the conception or the fertilization process. Next, we have cellular transport. Your cells need to maintain the balance. Okay? So they transport stuff so that they maintain homeostasis, a balance of conditions within the cell and against the outside of the cell. So the plasma membrane is what keeps the inside of the cell distinct from the outside. So this plays a key role in cellular transport. So its 2 layers are made up of phospholipids, a phospholipid bilayer, and your phospholipids have a hydrophilic head, meaning it loves to interact with water, and the hydrophobic tail, meaning it hates water. So what happens is that because of these properties, your phospholipids will arrange into a sandwich, a bilayer, where the heads interact with the water and the tails hide away from the water. So we have the hydrophilic heads interacting with water inside and outside of the cell, and the hydrophobic tails in the middle so that they're protected from water. Now, this lipid bilayer is selective and semi-permeable, which means that it's basically letting some stuff in and keeping some stuff out. Okay? So selective permeability, again, some substances can easily pass through, while others need some help to pass through the lipid bilayer. So hydrophiles, your polar substances, cannot easily pass through, while your hydrophobes will be allowed to enter the enter through the plasma membrane through the lipid bilayer. So it can just literally pass through your small hydrophobic molecules. Now, this kind of passing through the lipid bilayer is called passive transport. It's the movement of substances without the need for additional energy. It just happens naturally. So it happens along a certain concentration gradient. So we call this diffusion. It's the passive transport from a crowded area or high concentration to a spacious area, low concentration. So that's the general nature of things. If you're in a crowded place, you'll want to get some space. So that's diffusion. So again, it goes from high to low or down the concentration gradient. So it's from high to low, down, going down. So diffusion will continue until it reaches a point of equilibrium or the absence of a concentration gradient when the concentration is relatively equal across all space. So stuff still moves around, of course. It's just that it's moving around at equal rates to different places. So there's no net movement. That's what we call it. So stuff is still moving around, but now it's random. Instead of going from a specific direction, from high to low concentration, there's no high or low concentration anymore. It's just moving around randomly. Next, you have facilitated diffusion. It's just the same as diffusion. Now there's the help of transporter proteins. We have the channel protein, which is literally just a hallway, literally just a tube. So it just provides a convenient path for hydrophilic molecules to diffuse through. On the other hand, we have carrier proteins. Instead of a tube, it's a protein that waits for a specific molecule and then changes shape so that the molecule gets yeeted to the other side. So your carrier proteins are transport proteins that change shape to transport specific substances across the membrane. So if solutes or solid things that are dissolved in water can't pass through the membrane, how does the cell reach equilibrium? We can also have osmosis, or the movement of water across the semi-permeable membrane until equilibrium is achieved. Or in other words, it's water moving to balance the concentration. So it goes from low solute concentration to high solute concentration. So again, from the matabang to the maalat part. Okay. So again, basically your osmosis is water moving to balance the concentration. We have 3 main happenings. We have the hypotonic solution, where water enters the cell. isotonic solution where water just goes in and out of the cell, and the hypertonic solution where water leaves the cell. So let's see why that happens. In hypotonic solutions, if you compare it to the sample, it has a lower solute concentration and a higher water concentration. Cell, because there's more water in the solution rather than the cell. So again, what happens is that the water goes into the cell. So in animal cells, it swells up until possibly bursting, while in plant cells, they swell up but doesn't burst because of the cell wall. It instead becomes turgid or hard. In isotonic solutions, these have equal concentrations with the cell. Again, it's not the water and the solutes being equal to each other, it's these concentrations being equal to the concentrations in the cell itself, or in the cell sample itself. So in the animal cell, that's essentially normal. In the plant cell, it becomes flaccid or soft because there's not enough pressure. In hypertonic solutions, we have lower water concentration. In your hypertonic solutions, these are the super maalat solutions. Hypertonic, sobrang alat. So your animal cells would shrivel and shrink and they may die, while your plant cells will undergo what we call plasmolysis, or the wilting process. So the cell membrane is actually separating from the cell wall. That's why it's extremely dangerous to have extremely salty soil. So your hypotonic, isotonic, and hypertonic solutions, this is the main effect that they have on your different types of cells. Okay, so this is just the summary slide. And what we've been discussing earlier is all passive transport, but sometimes you really need to have energy in your transport process. So this is called active transport. So this requires energy in the form of ATP because it transports materials against the gradient from low concentration to high concentration. So it goes against the gradient. It's an uphill battle. So an important example of this are ion pumps. They actively transport hydrophilic ions across the membrane. So again, they transport polar ions against the gradient. So it's basically this, it uses ATP to transport stuff to the other sides. This is actually really accurate, the correct number of molecules and the correct number of ATP. This is your sodium potassium ion pump. So proteins can only transport so much. If you need to transport in bulk, you have bulk transport, which involves vesicles in the plasma membrane. We have two basic types: endocytosis, taking things inside the cell, and exocytosis, making things exit the cell. And endocytosis is just swallowing up stuff using the plasma membrane. Exocytosis, it's vesicles fusing with the plasma membrane and then yeeting stuff out. And that's pretty much it. Everything about cells in this lecture video. Congratulations if you've made it this far. Again, make sure you've taken notes and make sure you study your notes and refer to them during our activities. Thank you for listening, and I'll see you guys next time.
Party over.